Good evening, everybody, and welcome to our first in-person science talk in our new building here. We are pleased to have two of our main scientists here. We have Dr. Kathleen Pig and Dr. Melanie DeVore here, and they have not been here since before COVID, so we're really happy to have them. Plus, they brought some grad students, and so we have Daniel Moses, and Daniel may be familiar to some of you. He pretty much grew up here, and I heard a little while ago that he found his first fossil at 2 and cracked his first fossil open at 5. So I bet you don't know who told me that. And then we have Annie Weaver Bryant. I told her I'd forget her name, but Annie Weaver Bryant, who we're really happy to have here, and this is Annie's first time here, so we're hoping that she will start a long relationship with Stone Rose. So welcome to you all. We're really excited to have you and to have this talk. I wanted, before I give the mic over to Melanie, to Kathleen, I wanted to let you know about our fundraising events that are going on. We have this great new building that you can see. I don't know if, I think most of you have been to our old building and know how cramped and small that was. This building is much bigger. It's like 6 times bigger. You can walk through it without fearing that you're going to knock over necklaces or toys or something. So we have a lot of plans for this building, and we have started on a capital campaign to fund some things. So our fundraising is going to pay for the building, secure the building, make some structural improvements, and then we're hoping to have a lot of really cool displays. So we'll be starting that. And the other fundraising that we are doing is our end of the year. It's almost the end of the year. I know it's hard to believe, for me anyway, that it's almost the end of the year, but in the next couple of weeks, we'll be starting our end of the year campaigns, our operation. And so that does everything from, buy pens and pencils to maintain the site, pay for our excellent staff, Travis and Andy, wherever he's over there. And so would love for you guys to help us, and if you are able and willing to do so, I left some brochures in the back. They look like this. They're on that table back there. The website's on there. It's really hard to read because it's in the class, but it's on there. And there's a phone number there. So if you're interested in partnering with us for any of those, give us a call or talk to us on the website. You could talk to me or Michael or Travis. Yeah, we'd love to partner with y'all. So with that, I will give you Kathleen.
I think I have
Oh, you have one. Okay.
am I okay? Okay, we have been coming up here to Stone Rose, me for the last 17 years, Melanie for the last, I believe, 16 years. And before that, we worked with Wes Weir at the Burke Museum in Seattle for many years. And it's been our real pleasure and joy to be able to work on the fossils from here. We published, 20 or so papers on all kinds of topics. And we've had the joy of having our students, our undergraduates and our graduate students work with us as well. And so it's especially nice to be able to bring 2 graduate students with me. We come from Arizona State University and the BioKIC, which is Biodiversity Knowledge Integration Center. That's our official name in the School of Life Sciences. We're off campus and we're in a neat little building where we study all kinds of things like bugs and plants and fossils and things. And so we've got 2 of my students, and then Melanie, who comes, of course, from Georgia College in Milledgeville, is going to present as well. And so my first student is Annie, and second student is going to be Daniel, who most of you know, and then Melanie is not my student, but she's my dear colleague.
Very good.
In what is it called? in zealous research.
Okay.
That's what it is. Okay, so please, Annie.
All right.
Annie's going to talk about a plant that she's working on in the rose family, and she's been looking at flowers and things. This presentation that our students gave at the Botanical Society meeting back in the summer. Okay.
Oh, thank you so much. That's loud. All right, hi everybody. So today I'm talking about fossil Physocarpus. Another name for that is ninebark. And as you know, it's out of this locality. So I have a slide here kind of showing the locality. There's Boot Hill, and we're in the bottom of the Okanagan Highlands. I know you probably already know all of that, but you know, actually, does anybody in the audience want to talk a little bit about their experiences here? Some of your history, since I'm brand new. Go for it.
My family and I have collected fossils here since the year 2000, and it's been my joy to watch my son grow from a 6-year-old to a 28-year-old on site, and go from a yelling, screaming tot to a 28-year-old machine that can harvest huge numbers of fossils in an hour. But this hobby of ours has grown into a wonderful lifelong passion, both for my wife and I, and for my son. And it's a wonderful thing that Stone Rose has here. It's near unique in its conjugation of lay people like myself and the science community, and it's a wonderful thing and should be appreciated for what it is. And it's my joy to see it move forward.
See, I think that's absolutely amazing, because the cool thing about Stone Rose is, like, you have these 50-million-year-old fossils, but you also have the history of the people here. The fact that your son got to grow up doing that, that's amazing to me. I love that. We kind of advanced forward on the slides a little bit, sorry.
Scroll the other way.
I didn't. There we go. Okay, no worries. Let's see. Nope, it's not letting me. Okay, so there's Physocarpus on the far left end there, and it's native to both North America and parts of Asia, but it's mostly planted ornamentally. You can kind of see the growth habit of the plant. And then the photos in the middle are the buds of the actual flower. You can see that it's segmented into 5 parts. So that's what you can kind of see. It's here, but it's also on the fossil specimen. And then on the far right, we have the flowers, which have numerous stamens, both extant and then in the fossil. And something cool, I'm going to point again. You can see this right here is the pistil, which is still attached to the fruit. And it does the same thing. It's really hard to see right here, but it does the same thing in the extant fruit. And over here, you can see the D-shaped carpels. Kind of compare there. I think it's amazing. I've been having a fun time looking at it, and kind of the fact that we have the bud, the flower, and the fruit. We have all of these stages to look at. And I swear I'm not touching the mouse. I just CAT scanned some specimens, so that's what I'll be looking at next, and hopefully be able to publish in the near future. I know I went through it kind of fast. Does anybody have any questions?
You might mention that that's a millimeter scale there.
Oh yeah, the scale is a millimeter.
Is this genus known from, or this species known from other localities from different ages?
I'm not actually sure. I've just started looking at it. I know that there is extant, it's native here. There's Physocarpus malvaceus, I think I'm saying that right. And we think there may actually also be a leaf, but we're not sure on that yet.
Thank you.
It's hard to tell in the slides, but how similar and how different are the current ninebark species that are here compared to the fossilized species?
They're actually quite similar, and the color is a little bit hard to see. Some of the differentiation, like this one, these pistils still being attached, that's one of the biggest things.
Are those sizes close?
Yeah, they're quite similar in size.
So I know we've had a lot of things that we've called Gordonia fruit over the years that look similar. Are some of those now going to be kind of migrated towards Physocarpus?
That is a good question. Kathleen, would you say yes? Yeah.
Yeah,
Okay.
most likely. Yeah, this is a fairly common fossil that we find a lot of.
Yeah,
over the years.
it's there. We have several. Okay. Anybody have any other questions? All right, I will hand the floor over to Daniel then.
When is your paper going to be published?
That is a great question. I will do my best to do it as fast as possible.
Good girl.
Thank you.
Get this all set up. Let's see. Slideshow from the beginning. Here we go. Okay. Hi, and welcome to my presentation on the fossilized Miocene midge gulls from Washington and Idaho. Thank you from Mary and Kathleen for introducing me. My name is Daniel Moses, and I'm a master's student at Arizona State University under Dr. Kathleen Pick. And for today, I would like us to go a little closer. Instead of going to 49 to 52 million years ago, I'd like us to go to about 15 million years ago to the Miocene epoch. And particularly, I want to focus on two localities. Okay, there we go. Oh, I guess we're now. Do I have to scroll? There we go. Okay.
Do you have to click or scroll or what?
Okay. So the two localities is Yakima Canyon, which is located in this region right here, and the other locality is located in Emerald Creek, which is located in south central Idaho. So to kind of give a little background, this is the time period we're looking at. It had the last gas of warmth before thermal deterioration, so this was a little warmer than kind of what was going through before it reached a much more colder point in this particular history. If we were to go back 15 million years ago to both sites, Yakima Canyon flora will be much more something akin to Georgia, Louisiana, and Florida region. A lot more bog-like, having bald cypresses, pines, sweet gum. Something like I said was accustomed to southeastern U.S. today. Emerald Creek, however, was vastly different. The site was located in 15 million years ago in a giant lake, and most of the material we find floated down to the bottom, and that's what we collect. It was drier and colder than it was today. Was colder and drier than it was during the Miocene. It was a lot more wetter and slightly warmer. So what do these two localities have in common? Well, they have this tiny little guy up here. It's a bald cypress gall midge, and this guy is so small, he is roughly 2 to 3 millimeters in length and 1 millimeter in diameter. He is a very tiny guy. And what his favorite thing to do is to basically go after bald cypresses and create a little tumor-like structure on it. So as you can see right here. And from these ones, it can lay its eggs, it'll have nutrition and food. Basically, it bites into the bald cypress, injecting a bacteria into it, causing the plant to panic, thinking that it needs to scab over. And this allows the structure to start forming. So essentially, they're living in a botany-sized tumor on the tree. Once it is completed, they basically start laying their eggs in the larva chambers, which can be on either side of the gall. And once they hatch and form into larvae and the pupa, they basically start consuming and eating towards the outer edges in their best way to get nutrients and to avoid predators from either from bees or ants or anything else that might go after them. So for all intents and purposes, it is considered to be parasitic, but these little guys, the galls themselves only grow to roughly about 20 millimeters in length. So unless there's a ton of them on the bald cypress, it doesn't cause immediate issues. However, too many of them and the taxodium will start to droop onto the ground, and this will cause ground rot and other issues. But for all intents and purposes, it's parasitic, but is relatively harmless unless in large numbers. So this is some of the fossil samples we have collected from Yakima Canyon and Emerald Creek. The Yakima Canyon, as you can tell, is more three-dimensional, but the Emerald Creek is a compression fossils. So why is this important at all? Well, I explained earlier that the gall midge itself was extremely small. It's tiny. In most cases, in compression fossils or in the fossil record, it doesn't show up. It either gets eaten or doesn't go through the process quite right. The only time we've ever been able to see it is within the Baltic amber samples that have been collected either in Russia or elsewhere. So this is the only evidence we have to their existence within the fossil record. Within both paleobotany and paleoentomology, it's a bridge between two of the respective fields. Within paleoentomology, they mostly focus on trying to have the complete insect, the wings, the body, the antennae, everything about it. But with this case, we cannot find it because it doesn't either compress right or it doesn't get preserved right. Within paleobotany, it's something of an insect-plant relation, but it's mostly an insect initiating the plant relation. So this is a way for us to acknowledge the existence of an insect that may not necessarily be considered because it doesn't fit into either field respectively, but it is a bridge between the two fields. So this is what has us pretty excited, is it allows us to acknowledge insects that may not have necessarily been seen in the fossil records as having a place within it based on the habitat and the remains they leave behind from either feeding or home building. All right, and I think that's the end of my slide, if I remember correctly. Yes. So is there any questions for the audience? Go ahead.
Is the Emerald Creek material from Clarkia, the Clarkia flora?
Yes, it is.
Excellent.
And the Yakima Canyon, as stated, is from Central Washington, so near Yakima. Yeah.
What's the matrix of the Yakima Canyon material that it's so three-dimensional?
It's mostly a kind of, ooh, I have to actually look a bit more into that. Silicified? Thank you, yes.
It's silicified. And what happens is these little items come out, so instead of solid rock, the rock weathers out, and so we get these little items in the ground like that.
And recently we have done CAT scans on them. Within the compression fossils, we haven't found much yet, but within the Yakima Canyon's three-dimensional fossils, we have found evidence of at least catalyst or crystallization inside of it, implying that there might have been some hole digging at the center core. So we're having hope that this kind of goes further into confirming the gall midge and the gall midge's existence within at least the Yakima Canyon flora. All right. Any other questions? Oh.
Is there only bald cypress?
For this is the interesting part about the gall midge. So there was debate about whether or not there might be a connection with the spruce midges by any chance. But however, recent activities have kind of confirmed that gall midge on Taxodium only goes after the bald cypress alone. The spruce ones will only go after the spruce, for example. So for right now, that's all the confirmation we have on it. So yeah. And so for right now, as far as I know, this is the only galls located within both sites. Right. Any other questions? Yes, Michael.
Daniel, would you like any specimens of extant spruce galls?
Ooh, I would love some actually.
I have tons.
Okay, awesome. I'll take a look at them.
So did you collect only from the main racetrack site, or did you go up to Dr. Remmer's locality?
Ooh, actually, that's an excellent question. We've actually collected from both. We found gall midges within, the galls on Taxodium at both the racetrack and at Bill's actual direct site. So we found that at both localities. Some of these pictures were taken from the Idaho State University's collections that they were willing to have us look at. But yeah, other samples I've collected directly from Bill's site, actually.
Yeah. Yeah, we've been there 4 times.
Excellent.
It's a terrific locality.
Oh, I know. It's lovely, isn't it? Especially the insect.
Lots of taxodium there.
Mm-hmm. It's an excellent site, actually, and one of my personal favorites. So yeah, alongside Stone Rose. Yes, Judith in the back.
I was gonna say, you might mention that right now at Bill Ramber's site, you and me are trying to find one that's, down in the DNA pit, so that it's not quite so compressed, and that you're actually gonna be able to find, maybe a midget.
We're hoping so, yes. Like I said, until we actually find it, that's one of our biggest things we're looking for. So yes, we are trying to dig further in to see if we can find a more complete one. We sent them to Austin, Texas for scanning, and for right now they're appearing a little bit blank, but about 4,000 images of them. I'm still going through them for a little bit, so it's still a little bit of a process. Yes, Michael.
Have you, are there other Miocene age sites that might have taxodium and associated galls?
I believe if there was to be taxon there, I do believe there might be, but for right now, I was just focusing on the Yakima Canyon Emerald Creek because that's the one we have direct fossil remains of for right now. I think if I start expanding out, I might be able to find more, but for right now, just focusing on the Emerald Creek Yakima Canyon was the idea.
As Dr. Rember, or yourself, used his acid technique to actually lift the fossils off the matrix for the galls, or have you discovered anything that you could use for that?
We had discussed it as a possible way, but the problem here is that, as you see with the sample with P33, for example, sometimes when it splits in half, it doesn't quite come out quite right. So, and the bad part is, while we have found samples there, it's one of those things where, do we take the risk of destroying the sample or anything else? But if we found, but as my mother pointed out, Judith in the back, and I'm sorry, Mom, I'm calling you Judith, so that way people know who you are. If we find a complete sample, that might be a possibility in that regard. If we could extract it, then we might be able to get something very similar to the Yakima Canyon galls in regards to 3D scanning. So, or the scanning for CAT scanning. My apologies. So that's our hope, anyways. Right. Oh.
Where in the canyon is it talking about?
Really up. Way up.
Any other questions by any chance? I'm happy to be here all day if you guys want me to. All right. I will go ahead and turn this over to, let's see, my other advisor. Are you up next, Melanie?
Yeah.
Okay. I will turn over to my another lovely lady, Melanie, over here. And yeah, I will
Get rid of
exit.
your.
Let's see if I can actually get, oh, and show. All right. All right. Let me go ahead and
Do it this way. Okay, thank you. I'm scrolling and getting this up. There's kind of a method to our madness in the organization of these talks tonight. For many of you, you know we give talks at meetings, right? What you may not have known, that the presentations you just saw were things that were called lightning talks that were given at the Botanical Society of America's meetings this past year. And it was your first talks. So for first presentations, those are really extraordinary jobs, and they really represent Stone Rose outstandingly. And one of the things, too, is we want you to have a chance to see authentically what we present to the public. And frankly, for the amount of years that I've been coming here and working with Kathleen Pigg and students from both Arizona State and Georgia, we owe our career and an awful lot of our career to so many of you that are in this room, because you made this site so extraordinary by all the work that you've done here. There's no place that we can imagine that keeps an active fossil site that every single year people are bringing things in. Usually what happens is a group of people from a university get in vans, pack tents, and they go somewhere and spend about a month. So what you're able to do is that you have presented an extraordinary resource for research because you keep bringing in the specimens. So anybody from a 2-year-old, right, to a 102-year-old can contribute to the process of science. And I think now more than ever, it's important for people to realize that citizen science And the public's input into science is one of the most important things that we all need in order to progress further. So give yourselves a round of applause for that. All right, now for some bigger creatures. And again, this is actually a talk, one version I gave at the Geological Society of America in Cincinnati. It was a sectional meeting, Southeast section. So some of you in this room are familiar with this GSA, and many of you have already been here, and you've done a great job, in fact, hosting a booth to entice the geologic community to bring schools here and their classes here, and to incorporate Stone Rose into field geology courses throughout the Northwest. And I think that was just, and it's been, people who have visited here have been extremely impressed. How could they not be? Okay. So the other place I gave this was at the same meeting that Annie and Daniel had presented, which was virtual and in person, thus called hybrid. And I want to tell you something that you should be very aware of. You could attend the meeting in person, that's great, but we want to encourage our communities to offer them hybrid. And in the case of the Botanical Society of America meeting, anyone in this room could register for $50, and you could have access and participate in the meetings and participate and see the talks, which includes the paleobotany talks. Also, it includes the education and outreach. So for two of your big strengths in Stone Rose are represented at these meetings, and it would provide you a great way to make some good connections with other facilities, other interpretive centers, other arboreta, and other groups that are doing citizen science, but nobody does citizen science like Stone Rose. All right. Okay. We always think about plants influencing animals, but we never think about the fact that the animals actually influence the plants. And you know, when you look at the midges and gulls that Daniel presented, you know, you become aware that there's so many relationships plants have with so many organisms. And in fact, you know, they are the basis of the food chain. And so one of the things that we want to do is, how did all of these animals kind of get situated? How are they fitting into the forest? You see these reconstructions all the time. And some of them are designed to show how many different types of fossil mammals are dinosaurs, and you would see 100 different dinosaurs. But how did they actually exist? How were they actually influencing the environment, and how were they actually influencing the plants? So if you came to one of the questions that I'm really interested in, and is, what did Stone Rose, if you came to Republic and you were looking at Lake Republic, and you were looking at the forests around there, how did the animals fit into the picture? How were they influencing the plants, and how were they actually living and interacting and impacting the structure and the function of these important forest communities? All right. Okay, and again, I want to show you authentically what a talk is that we give. And in this case, I want to show you that most of the time when we're trying to make these comparisons, we use what we call a modern analog. All right? We don't have a time machine that we can take to Boot Hill and get zapped back to the Eocene. And we also, and most people who do these kinds of studies where you look for a modern analog often study things that are not that old. So over here is actually a gentleman, he's an Michael Kowaleski. He was at the University of Florida, and he shows up in the Bahamas when I'm there, and what he does is he takes students and they look at the actual deposits that are in rocks that are about 100,000 years old, and then go and look at all of the processes that were responsible for depositing them in environments, and have this model, this modern analog that they can immediately compare with the rocks that are there. And this is not an old idea. It goes all the way back to Hutton in Scotland, you know, the uniformitarianism, that the past is the key to the present, and the present is the key to the past, but the past is also the key to a present too, which is a whole nother bag of issues and things that we talk about with conservation paleobiology. That's not the right way to do that. Okay, you don't have to worry about all this, but just think about when you crack open a fossil on Boot Hill and you look at that fossil, think about all the different processes and things that have to happen for that fossil to be preserved. And one of the things that I've been interested in are these ecological processes. What can we look at as far as here's the biosphere, and you have paleobiological samples, better known as fossils. Okay, they couldn't give people PhDs unless they came up with some of these big words. And this is what I want to focus on today. So how can you look at fossils and get clues to what's going on in the biosphere? And when we talk about mammals, fossil mammals, one of the key types of fossils used are teeth. And it's kind of the classic things. And many of you, you're so familiar with Carolyn Stromberg's presentation because so much of what she does with phytolith links directly in with teeth and looking at teeth and just gleaming information from teeth about the plants that the animals were eating. The thing that I really like, and most of the time that I'm focused a lot on, is the plant response to herbivory. Okay. Well, where do you find a modern analog? And people had asked, how did you stumble upon doing this? And so I was one of those kids, like when I was like 7 years old, Mom brought me in and says, I want you to watch this National Geographic movie or National Geographic documentary about this lady that studies gorillas. And so, you know, the classic Dian Fossey gorillas was in Virungas, and if things that happen as a kid, think about the things you do with 7-year-olds here at Stone Rose. What happens when that sticks in your mind? You know, what donors and people who give money and support to Stone Rose are doing is they're sparking and putting in the seeds and ideas and things that get stuck in your mind that someday are going to really bear a lot of fruit. You know, so for many of us, we grew up on National Geographic. We had television that brought things into our living room that we never knew existed in the world. We didn't have Google, all right? So we had television. And it turns out that I thought, well, you know, I'm at the age where I can afford to splurge, and I'll just go on one of the programs that the Fossey Fund has. And besides, the Fossey Fund is in Georgia. It's actually in international offices at Zoo Atlanta. And I was out with the director of the Fossey Fund looking at plants, and it struck me just how similar, you know, what's it like when you have a warming environment and zonation, and what's it like, how do big animals actually exist in forests? Did you ever think about that? We have all these big animals, and we talk about forest. How do you fit them in? How do they habitat? How do they alter the habitat? How do they make the habitat favorable for them? And so it turns out that the Virunga Mountains is one of the best places that you can go. And I started working with undergraduates at the University of Rwanda that were involved with the Dian Fossey Gorilla Fund's memoir program. And so if you want to learn more about the fund, and I always have to say this, you can go to their website and you can learn about their gorilla conservation efforts to help people and save gorillas, which is their catchphrase. All right. A region of the world you all know and love and are very familiar with, the Okanagan Highlands. So we're going to come back to the Okanagan Highlands. And one of the things, too, is one of the valuable things about Stone Rose, and I point this out when people in the Fossey Fund look at the fossils from Stone Rose, which they do, believe it or not, is you can collect the fossil, you have the collection here, but you can also have the context of walking up the hill and seeing the actual outcrop. And so I can't think of anywhere else, with the exception maybe of places like Florissant, where you can see the site and have the whole context of the fossils in one place. This is really an unusual feature here at Stone Rose. And so we know we had a fossil lake where leaves and insects and fish floated out, sunk to the bottom. They were well preserved. Here's one of George's goes, who's somebody we dearly love and is a frequent visitor here at Stone Rose and has done some really lovely work and has published on the geology of this site. You're familiar. I mean, you can look and go out on the hill and you can see, you know, starting out, where do you find most of your fossils at? It's not at the top, it's in these shales. Okay, so these, all right. Question is, I always like to think, who's out there? What were the big creatures that were out there, the more fantastic beasts that occupied the Eocene landscape? And the latest early Eocene, this whole age that we have here represented in the Republic fossil site, there was an incredible period of time for the diversification of mammals. And many of them were known from Asia, North America, and Europe. And there's been work, of course, of the mammalian fauna. In fact, there's been now work with the birds. I think Bruce Archibald, who should have shown up, is on his way. I hope they let him across the border. Did work with the birds. And what Bruce and I found, just we love this. Bruce, one of those birds that Bruce found is called a mouse bird. So the only places you found mouse birds today is in Africa. And in particular, you can sit in the Virungas and see mouse birds eating fruit. So it's kind of like it in life. You have an unusual connection between two different geographic places and two periods of time. So these Okanagan highlands, we know that they were associated with densely forested habitats. And you had things like a tapir, and we're familiar with tapirs now from South America and in Asia. And of course, I love this, a hedgehog. You know, everybody in Britain thinks they have the market on hedgehog. Well, you've got your own hedgehog here in Republic, Washington. And also you had some of these other larger elements of the mammalian flora, like the Brontheridae. And there's distinct differences, as you know, between these upland and lowland Eocene floras. Here's the Republic flora. You're very familiar with that. Needs no introduction. And then places like the Green River. And so in kind of a more smaller series of lakes that was in this graben. And then here is kind of these very large inland lakes. I mean, I always think of them like almost like about the sizes of Great Lakes, or even bigger. And for those of us that, if you can tell, I have an unusual accent from being from Ohio, you know, in an area of the Great Lakes. And these lakes behave and act really differently from lakes that you would see here in Washington State. And so I always think about the big difference that you have in the nature of the Lake Republic and the Green River. All right, here we go. Here's for Daniel. Here, this combines Daniel and I together. We're bonded in this particular slide. I love this. It's just, I think it should just be called herbivory, because here you can see all the leaf damage. So we have the little herbivores, and then we have the apex herbivore, one of them, which is a mountain gorilla up here. This is the silverback watching everybody. And this is an image that comes from Conrad Lavandera's work, or other people, sorry, yeah, got it right, work. And it's, look at the bottom, here it's green, here's the plants, all right? And then you look at invertebrates. And then if you look at the number of vertebrates, we don't know that much, and just think of them as right here, these balls that are vertebrates. We don't know that much about the way the vertebrates really interacted with the vegetation. If you think about it, what were those relationships are. You're all familiar with the relationship with grasslands that Carolyn Stromberg talks about, and that's an easy one to follow. But how do you kind of begin to unravel and decipher some of these some of these relationships when you get into the older periods in the time, the earlier portions of the Cenozoic? So, I mentioned images. So, for example, do you think a tropical highland wetland looked like this? How many people think a tropical highland wetland would look like this?
Tropical, yeah.
Okay, all right. So you think about the way the plants ring and structure, the structure of the vegetation, the zonation that you would find. And so if you look at one in the Virungas, this is what it looks like. Okay. looks quite a bit different. Okay. And we might think, well, in that previous slide, you were looking at a glade that was made up of this particular plant, Carex. Okay. It's a sedge. You know, some of you that you took your field botany classes, sedges have edges. That's what we're talking about here. And this is a stone rose fossil right here. Here are more additional stone rose fossils. And we find that we have the collection, the aggregates of the fruits and flowers, the flowering heads and spikes of Carex that are represented. And you notice about it, everybody who's been up on the hill, you found those pieces of leaf that looks like somebody mowed the grass and tossed it into the lake. And so think about how often you're out there digging and you find that. It's all over the place. It's quite a common element. So think about the aspect that how prevalent Carex could have been and these plants could have been in, and how they could have been a major zonation around the lake. The other thing too is we think about information in subhabitats and so forth. Okay, if you eat grass, you tend to have, and this again, I feel inadequate talking about this since Carolyn Stromberg is the expert here on this particular thing. You think about the high crown tooth in horses and the way that it gets ground down, grasses. If you eat leaves and foliivores, in the Virungas, for instance, you don't have fruit. You don't have a lot of fruit. You have like blackberries, raspberries, the genus Rubus. But these animals that are herbivores are eating leaves. So think about in your mind how much nutrition and how significant amount of nutrients that you can get from a leaf. So if you look at a mountain gorilla, if you look at the silverback, he's not fat. That's all muscle under there. So the biggest primate on the planet is truly vegan. Okay. And what's interesting, if we go down with these habitats, you know, in woodlands, you have these brachiodonts. But what I think is always interesting is where did all these grazers come from? Did they just kind of appear magically out of Athena's head? They had to be some of them lurking and being in some of these subhabitats. So you could think of some of the precursors of some of the very important grazers had to have been somewhere. They didn't just appear. They had to have been pre-adapted so that when times came where the vegetation changed and grasslands evolved, they could move out there and go there. And here you have some in the Virungas. We actually had a buffalo paper that came out with the Fosse Fund with one of the memoir students looking at the shift in C3 and C4 grass consumption by these guys. So what's kind of cool here is look where they're at. You know, they can think about the weight of having groups of buffalo go through. Think about the trails that animals go through. And one of the things that's very good about the Virungas is you even have elephants. You have the forest elephants that kind of go in groups of 4 or 5. So think about when elephants and buffalo and large things move through the woods and the forest, they're creating pathways. So in fact, the pathways today that people follow when they track gorillas are ones that are created by these large herbivores. The other thing is they trample. So if you trample and compress and the surface here enough and the soils, you can create a favorable habitat for a lot of the food that you eat. The other thing, and getting back to teeth, is that there's wear patterns on teeth besides horses that can give you indication about things that animals are eating. In this case, we're looking at gorillas. This is a great paper that came out just last year. And there's distinct stages that a tooth wears during the lifetime of a gorilla. So close your eyes, think of the first gorilla you saw at a zoo. That's a western lowland gorilla. They eat a lot of stuff. They have a wider diet. And you can see that if you are eating fruit, you have a different wear pattern than the classic mountain gorillas that only eat leaves. So you can actually look at these wear patterns and actually get an idea of, was this a forested area without fruit as a primary source like the Virungas today, or was it a tropical area where you had more fruit? The thing we love is that plants respond to mammals. So we have fossil rubus in Republic. Rubus is also in the Virungas. It's one of the top 5 gorilla food plants. So through this, you'll notice there's Basoque. That is actually within the park. So here's the park here, and here is the outer portions of the park. So it's kind of like the Lorax. When you watch or read the book, the Dr. Seuss book, The Lorax, and that poor Lorax is stuck up in the mountains and just only has so much forest. This is how these animals live, today. So you have so many people, so many agricultural areas that are developed, and they're pushed into these mountains. So you can see here, this is within the park, and these are on the side of the park. And of course, you have to show a gorilla eating a stem. So here's rubus, it's eating the stem. And we think if you're eating the stem and you're eating parts of the plant's going to have some responses. And of course, if you want to stop eating all you have to do is bite into something that has a prickle in it or a thorn in it. Even when you clip your rose bushes, you know how it feels like when you grab them and you're not so careful and you get pricked. So, part of the thing is the plants are going to be responding by having these pricks. So we had a student, and what Pasteur did was he basically did all these measurements, and I just want to point you to this part of the graph right here. All right, so this is inside the park, here's outside the park. And so the way this plant is responding is, you see how they grab the stem? They produce more prickles, it's more dense, so they produce more prickles on the stem. The other thing you can do is you can force the plant can also change its shape of these prickles, and so that they're more effective of penetrating the skin of the herbivore. And so again, you wouldn't believe how much time this Rwandan student that took measuring and collecting these data. So sometimes it's really cool when a student gets really great results. And he did. And so what they found too is that you had within the park more straighter prickles and more curved. And we actually used some techniques that people who study hawks and velociraptors, the curvature, to tell the efficiency of the penetration, some of the geometry was used to and quantified that allowed him to do that. So he was really a creative guy. We had a lot of fun doing this project. So if you look at that curve, you could, if you're starting right here and you move that way, you're not going to hit the sharp end of the spine. But if you just grab this and it's thicker and a little bit more robust, you're going to get crashed. The thing that's cool about rubus is it appears that they can induce their prickles to become longer, more upright, and more dense. And that's because each prickle starts, lost the cursor here, starts, there we go, as a glandular hair. So here's this little glandular hair here, here's the little glandular hairs here, and look, and here's the developing prickle. So if you would look watch some of your blackberry or raspberry plants that have prickles, they start out with the young with a lot of these hairs, and then by being mechanically jolted, be induced to produce prickles, and sharp ones at that. And the question is, can we apply these kinds of data with an example from a fossil record? And so for many of you, you were familiar with the fact that one time we were showing leaves that had prickles, and we went and we had a digital microscope, and of course it was in the old building, the old Stone Rose Center, and we sat there and we had more fun looking for prickles on leaves. And people found them, and we used these data. So it was really fun to have people that were visitors to actually come in, spend time with us, helping us, and also the fact that they were finding them. And some people have an incredible eye, and some people, it's like, okay, All right, if you say so. Okay. So if you take a look, these look glassy, and that's because they are. So think about if you had a very narrow glass cone, and it was filled with an irritant. And so if I touch this edge right here, it breaks off. And then as my hand or my body part, whatever body part is touching the leg, arm, it would actually, the broken off part is sharp and penetrates the skin and releases this irritant. And immediately the animal drops it. So stop and think, if you're a plant and there's a big animal eating you, and think about how big these Cenozoic mammals have been or were. You don't have much time. You're not going to last long unless you can have a means of defense that's going to operate really quickly and have a big impact. And so for anybody nettles, just imagine taking a mouthful of nettles. I think you would drop it instantly before you would do much damage to the plant. And so again, another student, Alphonse, he's now in a doctoral program, I believe. He's amazing. And he went in and he did the measurements and worked, and he was looking at the trichome density. And what's interesting is right here, La Portilla, this is the primary member of the nettle family that is one of the top 5 or 6 food gorillas. Okay. So again, it has the highest density of trichomes. And insects are smarter than mammals. There you go, Daniel. So take a look. You know, numerous studies indicate that these trichomes effectively deter mammals. And people always say, well, how do you know it's not insects? Well, the insects are small enough, they just eat around them. So right here you see that trichome there, and then you can see the leaf damage as they eat around those. Okay. So we would think about it, if this is such a strong selection for herbivory, you would think that you would have it in the fossil record. And especially when you think about these are glassy kind of structures, you would think they would have high preservation potential. So what do you find in the fossil record? This is actually, we want to start just with some images that we actually showed you at first. So here was a herbarium specimen. This was part of Alphonse, and there we had the arrow pointing there. Here you have them here. Point them there, 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 there. And you notice they're not bent, they're rigid. You can see that preservation there. You can't mistake it for anything else. We also have the top of the leaf, and we have what could be bases of trichomes. And then there's the paper that came out. The other thing, it's really fun to start a story here at Stone with a talk, and then to be able to come back and say, okay, here's the paper. You're next, Annie. Yeah, we want to see FISA.
Your co-authors here.
Your co-author?
Your co-authors for this paper, where they come from.
Oh, yes, where they come from. So Alphonse is the student that has worked with the extant nettles. Winnie Eckert is a gorilla behavioralist. So she has been the research director at Karasoke Research Center, the Karasoke being the field station that Fosse established. Elias is at the University of Rwanda, so is Miriam. She at that time. I don't know where this person's from. Yeah, I think.
But this was to our knowledge, this is the first fossil paper.
Yes, this is the first this is the first paleobotany paper that anybody from Rwanda ever. Oh, cool. Yes, so you've got the first one. And so the best part about this story is I showed this particular picture to Alphonse and Dio, who is one of the staff members at the Fosse Fund. And I thought it would be really fun because we had these things and we thought they were rubus leaves. And all of a sudden, through Alphonse's project, the image just crashed back to me, what they were. And I showed this particular, or at least this particular leaf. This is, again, a Kathleen Pig plate. So give her a shout out for her ability to put these plates together. I showed this leaf and I said, Alphonse, Dio, what does this look like? Oh, that's La Portilla. So, and they said, is that in mud? That's weird. I've never seen one like that. Is that from a mud puddle? And it's like, well, no, it's from almost a 50 million year old fossil site in Washington State in the United States. Okay. So it's just really a pleasure to be able to see. I mean, think about this, the potential that we have fossils that were collected by people here in Washington State and studied here, you know, linking up and being a tool to collaborate with people who are in Rwanda is just kind of a fun connection, you know. And I think, and we never would be able to do this, because let's stop and think. Remember when I first was starting out this presentation, I said, the thing with Stone Rose that makes it such an outstanding site is that the people, everybody comes every year, everybody's collecting. You get more and more and more. Think about how many leaves you have to look at until you have the right split that shows you the trichomes. And so this is not possible unless you have sites like Stone Rose, where you have lots and lots and lots of material. So every...
As you go through different years, as you look at when, because you all record what year things were found, you go through different pockets of different plants too.
Mm-hmm. Yeah. Okay. So, and this would be a typical end of the line of a presentation that I would give, you know, with the conclusion. Use of teeth as a dietary proxy, and that's always going to be there. And they use isotopes, as many of you know, and again, all of the work that Carolyn Stromberg does. But we are really interested in how to develop a finer proxy for mammal herbivory from the fossil record. And this is in its infancy, if you stop and think about it. And this is the fun. We had the Rwandan Development Board that allows all each student. Here's what the Dian Fossey Gorilla Fund Memoir Program is remarkable. They take an undergraduate student, so you get to 10 at this last time. You got the 10 best botany students. And I can remember, well, would you like to work with this? Work with, you know, the students here, and you, oh, they're the 10 best. Oh no, I don't want to work with the 10 best. No. And so they literally learned the whole process of how to write a permit. They have to come up and write a permit for the project. They have to tell exactly how they would sample. They have to exactly say how they're going to analyze data. They have to tell exactly everything, just as you would have to do if you were a established scientist. And they go through in data analysis. You notice there's a lot of data in their presence, so they learn all of the statistics and how to apply them. And they have to present. And they produce a document that would be a manuscript. So the manuscript that came out this year on Buffalo, that was a memoirs project that was turned into a publication in the Journal of African Ecology. Okay. Of course, the Gorilla Fund, Cleveland Metro Park Zoo. Cleveland Metro Park Zoo provides the money for the undergraduates to come. And for zoos that are, and again, one of the things that zoos do is promoting conservation. And it's kind of like the animals there are flagship, kind of, you know, banner holders for species to get people interested. So you can go to the Cleveland Zoo, and they have in their display in their 3 boxes for conservation for kids or anybody to put a quarter in to save their favorite animal. And so you think about the number of people that visit the zoo and put quarters in. And when I'm there, the times I've gone there, I always stand behind it and, you know, threaten the child to put the quarter in for gorillas. All right. And of course, here we always do the Stone Rose Interpretive Center. It's just as always. So this has just been really a pleasure tonight. And for people who, first of all, if anybody's watching, we don't always remember there's people watching on YouTube. Get out your checkbooks. You can do this, start writing some checkbooks. Better yet, tell your child, you know, whether it's a 58-year-old child or a 15-year-old, let's put the phone away. Why don't we take your phone to Stone Rose and you can take pictures of the fossils you find? because the ability and the things that Stone Rose can accomplish are astronomical. And with this new facility, it's going to bring in new opportunities for doing things that were never possible in the previous place that housed the collections and the center. So this is the time I'm going to be quiet, and I'll entertain a few questions, but I think everybody wants beer and food. Okay, and thank you. All right.
What about, maybe another thing you could talk about just a minute with Rwanda is how COVID provided us with a lot of challenges, but also provided us with opportunities to do more outreach and online interconnectedness of our seminars and things.
Oh yeah, one of the things too is everybody is so many people have all of a sudden. First of all, let me start out. I was in Rwanda in March of 2020, like 3 days before it shut down. And we also thought maybe this is the safest place to stay. And then immediately you go back and your whole way of doing things within 2 weeks was turned upside down, where literally the entire nation had to figure out whether you were teaching kindergarten or whether you were teaching quantum mechanics to your PhD level course at Stanford, online. And different formats, different teaching platforms.
In the same room,
yeah.
Each of one, little kid and parent.
Yeah, whatever. But one of the things that happened was, and I say this as somebody at the university, COVID turned education enough on its nose in a way that it's going to be more creative. We found more innovative ways to do things, and that the whole way that we teach and think about teaching is going to change in ways that it's going to benefit more people than ever, especially in higher education. And so it allows you to bring people who do real things, okay, into your classroom. And so what I would do is like, okay, I don't study gorillas. I study their interaction with plants, with students, and advise them, but that's not my main wheelhouse. But I do teach a class where where I was able to have the FOSSI Fund come in and have people who were who were staff members, who were research associates, interact with students in my class and tell them exactly what they did, how they would go into forest, and then they would learn, well, how do you handle gorillas during COVID? How is that going on? And one of the coolest things is there was the paper that was done about how the groups responded, how would COVID go through gorilla groups, and what could be learned from gorilla groups in COVID and humans and spread. And what was so cool is that the presentation and the information the students got in that classroom that day was released before it was released in a press release or published in the New York Times a week later. So, it's really incredible what you can do. And we've been able to do more things connecting Arizona State. Stone Rose, we've had, that would be really the best thing is thinking about, here's the other thing to think about with Stone Rose. How many college level lectures and material and concepts that could be taught from your virtual classroom? So the sky's going to be the limit in regards to, it really is. All of a sudden you have the total old school digging to the virtual classroom.
To the other things in the region that are interesting along with fossils.
Yeah, there's tons of things that can be done. Just the sky's going to be the limit, and it's an exciting new era here for Stone Rose because you have this incredible new facility. So write out checks. People out on YouTube, get your checkbook. All right. Any questions? Yes.
This is Elliot.
Hi, Elliot. I'm Melanie. I'm really glad to meet you tonight. Hi, Elliot.
You had a question about
Oh. Well, there's a lot of people that get big on eating meat too. Protein. So plants have protein too. So you don't think about plants having proteins, but plants, just like T-Rex got his protein from the meat he ate. Do you like hamburgers, Elliot? Yeah, well, you get your protein from your hamburger like T-Rex would get from his meat. But you also can get a lot of protein from plants. You don't think so. Think about it. So you can just get as much protein from your salad. Soybeans. Yeah. Is T-Rex your favorite dinosaur? Yeah. What's your second favorite one?
Probably Velociraptor.
I like velociraptors too. They're cool.
Show her your moves, Elliot. They kind of match your shirt.
We lost some of our clothes here.
Oh. Oh yeah. Elliot, come on up. Come on up here. Come on up. Come behind the podium. You got to come behind the podium. Don't get scared. All right. If you don't mind, I want you to lift you up so the people on YouTube can see you. If it's okay, Mom? All right. There's Elliot's boot. Yay. All right. There you go. Nice job, Elliot.
Thanks, Elliot.
Okay. So I guess that I can turn you back over. Here.
Yeah, so I guess that concludes our program. Thank you so much for all four of you for coming and giving this great presentation. And they'll be around for a couple of days, right? Yeah. So, if you're in the area and have more questions or think of something, is that okay? Yeah. Okay.
Elliot, will you help me find fossils? Okay.
And Elliot's 7, right? No, but he's 7 years old. Yeah, so you were the right.
I was saying, yeah, I want to dig fossils too.
Oh, well, everyone wants to dig fossils. So anyway, let's give another round of applause.